Welcome to Mohit Agricultural Tutorial. My name is Mohit Gupta, owner of Mohit Agricultural Tutorial. Today's our topic is lethal genes. We will talk about what are the lethal genes and what are the experiments done to know what are the lethal genes, right? So guys, in my previous video I have talked about codominance. So if you have not seen that video, please go to description, link will be given there, right? So without wasting time, let's start our topic, that what is lethal gene and what are the consequences of lethal genes, right? Let's start it. Okay. When we talk about the word lethal, lethal means tendency to die or death causing, right? So this means death causing. Death causing. How will these genes cause the death? Main question arises. What are certain conditions in which these type of genes will cause the death? Right. So when we will talk about these genes, these are not like normal genes. These are called mutated normal genes. Right. So when a normal gene in contact of mutation, right, when a normal gene get mutated, right, when a normal gene get mutated, directly converts into a lethal gene, right. These are death causing genes. These are the genes that causes the death of an offspring. As we know, I am explaining this topic in my series of genetics. So this is little related to genetics and inheritance, right? The individual that have, or the parent generation, if the parent has these genes, he or she will not die. That is known, right? The offspring that will be coming later on, that offspring may die due to these type of genes. That's why they are called lethal genes, right? So let's talk about what are the lethal genes. Straight away talk about what are the lethal genes. So to understand lethal genes, we have to understand an experiment, right? What is the experiment that I am talking about? The experiment was done by Lucien Cuénot, a French geneticist, right? A French geneticist did this experiment to demonstrate lethal genes. What was the experiment? Let's talk about what was the experiment very fastly. So the experiment was, he took two types of mice, say a pure yellow colored agouti mice, and crossed this mice with pure grey colored agouti mice, right? So the quality of this mice is, it is pure yellow color. Okay. So this mice is pure yellow colored and this is grey colored mice. And what did this scientist did is that he did the cross between the two mice to know more about it. Look, the quality of yellow colored dominant agouti mice is that it has the genes, its genes is lethal, right? So this has a lethal gene in it, right? So capital Y denotes lethal genes, right? This is the point to remember. So then he did cross between the two mice to see what are the consequences of the lethal genes. So when in F1 generation, this was parent generation, right? When we jump to F1 generation, so in F1 generation what happened is, these two mice right, when we will plot them in Punnett square, the four offsprings will be heterozygous. And one thing I must tell you, that this offspring, that is a heterozygous offspring, will have a quality. What type of quality it will have? It will work as carrier of disease, carrier. So it is carrier of, say, lethal genes, right? It will be work as a carrier. When two of the heterozygous offspring will cross with each other, they will work as what type? Lethal genes carrier, right? So what I am telling, I will demonstrate now. So this was F1 generation. That result we came with our 4 offspring. Each offspring is heterozygous, having certain qualities. The quality of that organism is of both dominant and recessive. Hence, it is semi-death organism, or we can say they will work as a carrier of death. Now, when we talk about F2 generation, these both are organisms, heterozygous organisms, are crossed, having both qualities, right? These are yellow mice. Again, to tell you, due to law of dominance, these mice will be yellow colored. All the 4 mice will be yellow colored. So when 2 of the mice, yellow colored agouti mice, are crossed with each other, when we will plot it in Punnett square, what we will get is both are heterozygous, they both have been crossed with each other. And look here, right? So as we can see, what do we get? What result do we get? The 4 of the offspring, each offspring has some qualities, right? So these are the 3 offspring that we got, or the 3 results. The first result, having a quality that it died, right? So pure homozygous yellow colored offspring that was having all the quality of this parent actually died. Why did it died? The main question arises. So this was, and the both, it was a carrier, hence it lived, carrier lived, and recessive, pure recessive, it also lived. Only one died. So scientists concluded that when a mutated lethal gene having organism is crossed with another organism that is natural or normal organism, then in F2 generation, the pure case of lethal genes may be dominant, may be recessive. It may be dominant also, it may be recessive also. That I will tell now when I will tell the kinds of lethal genes, right? So may be dominant, may be recessive, but the pure case will die in this case, right? If the pure case YY has quality of lethal genes, then this capital Y, dominant Y, will die. Or if the pure case of YY, say both are recessive, have the quality of lethal genes, then this pure recessive YY will, this organism will die. So this was experiment done by L. Cuénot. Hence the ratio what we will get over here is 2:1 instead of 3:1. Guys, it is very normal thing that Mendel said that ratio that must come is 3:1, but over here the ratio what came is 2:1. Why the ratio came 2:1? Ratio must come 3:1, right? So this was one experiment to demonstrate lethal genes. Now we will talk about kinds of lethal genes, that how many kinds. Okay guys, now we will talk about certain kinds of lethal genes, right? So what are the kinds of lethal genes we'll talk about? So lethal genes are generally 3 types, right? What are the 3 types? Let's talk. So the very first type is dominant dominant lethal, right? Dominant lethal gene. Second type of lethal gene is recessive lethal, right? And the third type is conditional. Conditional, right? So very first we will talk about what are the dominant lethal genes, right? So let's start. So when we talk about dominant lethal, what are these dominant lethal? So as you know that the genes, when we talk about, they are divided into two categories. One is autosome and one is sex chromosomes, right? But we will not consider over here sex chromosome, that I'll make another video. We will consider only one, that is autosomes. So when we will talk about autosomes, there are three genotypes. What are the three genotypes of that we have are, we all know that there are only three genotypes. So very first, fully dominant, AA, right? Second genotype, AA, and the third genotype is fully recessive. This is, in every case we will get only three genotypes, right? So when we talk about dominant, so lethality, the only dominant allele or dominant gene will show its lethality. Recessive never dies, only dominant will die, right? If the pair of pure dominant is there, then it is sure that this one will die, but there are chances of its death also. So we will consider both of them to be died, means dead, right? So they both will die in case of dominant lethality. So death, right? This one, that is fully recessive, this type of pair of chromosome will live because there is no dominant one in it, right? So this will live. So when we talk about percentage of death in this case, then in the percentage of survival is very high, very low, right? Percentage of survival is very low in this case, that is 25% of the case will only survive, and percentage of death when we talk about, that is very high, that is 75% of the offspring will die in this case, right? So we will say it less safe case, right? It is not very safe, it is very less safe case, because 75 of the offspring are going to die in this case, right? So this is dominant lethal gene, right? When we talk about dominant lethal gene, all the dominant, wherever we will find the dominant, the chances will be there that it will survive or die. This is so very sure that this will die. But this, when we talk about heterozygous case, there is little confusion that it will die or not die, right? So but when we talk about percentage of survival, we take both together, right? Wherever the dominant will be, that will die, right? Because over here the quality of dominant will be shown according to Mendel, that's why the dominant. Now let's talk about recessive. What are the recessive lethal genes? So when we talk about recessive lethal genes, right? So when we talk about recessive, right? Recessive lethal genes. So in this type of case, all the recessive offspring are going to die. Right. Let's talk about its genotype. Again, the three genotype that we have in all the cases that are homozygous, heterozygous, and what type? Recessive. Sorry, what type? Fully recessive, right? Homozygous case. So these are the three genotypes, right? So over here, what will happen? Only these two genotypes are going to die, going to survive over here, right? So survival, life is over here. In these two cases, the genotypes are going to live, survive. But in this case, death is going to come. There is only one case over here where death is going to come, right? So when we talk about percentage of survival is very low in this case, right? No, percentage of survival is very high in this case, right? How it is high? Because 75% of the offspring are going to survive, right? When we talk about percentage of death in this case, only 25% of offspring are going to die. So this case can be said more perfect, because in this case naturally the offspring that we will get are very much live offspring are 75%, right? So this is recessive lethal genes. Now let's talk about conditional lethal genes, right? So what we'll talk about now? Conditional lethal genes. Conditional lethal genes. So when we talk about conditional lethal genes, it depends upon the condition. Let us have and guess that a particular type of, say we will take an example of snapdragon flower plant. Example, snapdragon flower plant, that is anthrinum, right? So in this case, what happens is, it is little conditional type of lethality this plant shows. That I'll now when I will demonstrate, when I will give certain examples, then I will tell what type of lethality does this organism or this plant shows. The lethality this plant shows is, in certain conditions, temperature or atmospheric condition, this plant shows its lethality, but in certain atmospheric condition this plant does not shows its lethality, right? So that's why this plant can be said as conditional lethal gene, right? Certain conditions are there where it will show lethality, or else it will not show its lethality, right? So these type of genes are called conditional. Now let's talk about certain examples, right? Examples, right? So the very first example that I am going to give is of sickle cell anemia, right? Cell anemia, right? This is a disease over where the lethality is seen, or the lethal gene are shown in this disease. Let's talk how. Say it is a disease over where there are two of the genotypes. Say hemoglobin normal, when crossed with hemoglobin sickle cell anemia, hemoglobin sickle, right? So these are lethal genes having quality of sickle cell, and when they will cross, so what we will get is 3 of the offspring. What are the 3 offspring? That are HB, hemoglobin normal. 4 offspring we will get, but we will consider only 3 offspring over there. So hemoglobin normal, the very first offspring that will survive or will not have sickle cell anemia. Second offspring will be hemoglobin normal and hemoglobin sickle cell, that will be affected. The third offspring will be but affected, but the chances is there that it will live or not live, or it will survive or not survive, right? Affected, carrier, it is also called carrier, right? The third case will be hemoglobin sickle cell, hemoglobin sickle cell. Now this offspring that we will get due to such cross will be sure, you must be sure over here that this will be perfectly affected and will die, right? So death will be seen in this case. Pure sickle cell anemia offspring, it will be affected by sickle cell very large extent, right? Pure, every cell is affected by sickle cell over here. There is no chances of life in this case. So this is first example, that is sickle cell anemia. Let's talk about another example. So another example for such cases are example 2. Example 2 is beta thalassemia, right? But that is a disease. We must take another example. Say anthrinum, snapdragon flower plant. Snapdragon flower plant, right? Now over here the lethality is shown in this plant also. How does this plant show the lethality? Let's talk about it. Say when these are the 2 genotypes, say this is pure dominant golden color leaf, right? So leaf color of snapdragon flower shows the lethality, right? When crossed with say dominant green color leaf color, right? Green color leaf. Now these are the 2 plants over where this golden color leaf is lethal, right? So the 3 cases that we will get is AA, that is the very first case. Second case AA, that is carrier. Third case again AA, right? Over where this one will die, right? Because it will be lethal, and these 2, right? These 2 of the offspring will survive. So the ratio that we will get is 2:1, right? So these are certain cases in which the lethal genes are seen, right, naturally, and the ratio that we will get in case of lethal genes will be little different, right? The normal ratio according to Mendel was 3:1, right? But in case of lethal genes, the ratio will come as 2:1. 1, right? So this is lethal gene case and this is normal case, right? So this was the end of the topic. So guys, if you like my video, please share this video, subscribe to my channel, like my video, and guys, stay fit, and guys